Hey, it's Moser with an introduction to sex-linked genes. We're going to go ahead and dive right in with a little bit of genetics review. So, you might remember genotypes, phenotypes, and that genes are in fact sections of DNA found on a chromosome. Yeah. And when we're talking about them in genetics, we reference them with these letters to represent the different variations that exist, the different alleles. So, the autosomes, which is what we've talked about up until now, carry a whole lot of genes. And these are the standard genes we've talked about. They're carried on the autosomes, and the inheritance pattern is always going to be the same for males and females. Because there's no difference between males and females in who's got chromosome 1, chromosome 2, chromosome 3, chromosome 4. It doesn't matter. So we see the same inheritance pattern. We don't have to do anything special. You've done this before. Do you remember these? Punnett squares. Yeah, so for autosomal genes, we can put down the mom's genotype, the dad's genotype, do a cross like this, and we actually use the Punnett squares usually to figure out if a gene is dominant or recessive by doing test crosses, crossing multiple individuals and looking for the right ratios in the case of something that is a straight dominant recessive. If we cross two heterozygous individuals, we should see about a 3-to-1 phenotype ratio. Okay, easy enough. We can also kind of go the other direction with an autosomal gene and use a Punnett square to actually predict what percentage of offspring will have each genotype and phenotype. So, yeah, we should see a 1-to-2 ratio of genotypes and about a 3-to-1 ratio of dominant to recessive phenotypes. And that'll be the same for males and females because, again, these are carried on autosomes. But what about genes that are carried on sex chromosomes? Yeah, sex chromosomes do in fact have genes on them. Mostly the X, because the X is significantly larger. The Y doesn't really have, well, it's pretty small, so there aren't a whole lot of genes on it. But we can talk about sex-linked genes or X-linked genes as those genes that are carried on the X chromosome. Now, for individuals with an XX, females, they should have 2 copies of the X chromosome. Males generally only have 1 copy of the X chromosome. Well, that makes a difference. And we'll get to that a little bit later. First, let's look at how we're going to write and talk about these X-linked or sex-linked genes. So sex-linked genes actually have to be noted differently. So here I've got our sort of sample X chromosome, and I've written down the names of some of the genes that we saw on it. When we write a gene that is a sex-linked gene, we write this X with a little sort of, we call it a superscripted letter for the gene itself. And the big X shows that this gene travels on the X chromosome. The small letter at the top right shows whether we're dealing with the dominant or recessive allele of that gene. So the thing is that sex-linked genes are inherited differently in people with an XX versus people with an XY. And you know what? We can still use a Punnett square. I know you're excited. That Punnett square is going to look like this. So we've got mom's information here across the top, and she is X big G, X little g. She's heterozygous for whatever this trait is. And we've got dad's information down the side. He is X big G, Y. So what does this mean for their potential offspring? When we're tallying up the genotypes and phenotypes in an X-linked cross, we always separate our female offspring and our male offspring. So if we look at our female offspring, and that would be the top row, we have 1 X big G, X big G, 1 X little g, or X big G, X little g, and 0 X little g, X little g, which means 100% of their girl offspring are going to show the dominant phenotype. Now, what about the males? So the males are the bottom row, and it looks like we have one X big GY, one X little GY. So 50% of the sons of whatever parents these are going to have the dominant phenotype, while 50% are going to have the recessive phenotype. Okay, it's not so hard. Let's try one sample problem together, and this is actually from your worksheet, and then you can go do your practice on your own. So in fruit flies, I colored a sex-linked trait, and the gene is on the X chromosome. Red is dominant to white, so we would show these as X big R for a red-eyed gene, X little r for a white-eyed gene. Now, when we want to look at individual genotypes, a red-eyed female, well, she could be X big R, or she could be X big R, X little r. Either one of those would get her a red-eyed phenotype. What about a white-eyed female? Well, the only genotype she could possibly have would be X little r, because she would have two recessive genes. What about the males? So since males only have an X and a Y, they don't have two X's, a red-eyed male would be X big R, Y, and a white-eyed male would be X little r, Y. Okay, with all that in mind, let's actually walk through a problem. If a white-eyed female is crossed with a red-eyed male, what percentage of their female and male offspring will be red-eyed versus white-eyed? Well, first, it would be really handy to write down mom and dad's genotypes, so that white-eyed female would have to be X little r, and the red-eyed male would have to be X big R, Y. Okay, good. Once we've got those, we can set up our Punnett square. So this time I've got mom's information across the top, dad's information down the side, and I'm going to go ahead and complete my cross. So I'm going to tally these up, and remember, I need to separate my female and male offspring. So for female offspring, I've got 0 with an X big R, 2 with an X big R, X little r, and none with an X little r. So 100% of the female offspring are going to be red-eyed. What about the boys? Well, for the male offspring, I've got no X big RYs, but I have 2 X little RYs. So all the boys, 100% of them, will be white-eyed. That should get you started. Go ahead and finish your practice and ask questions as they come up.